discussing nucleic acids and the learning objectives. So I got a couple of questions on Friday that I just wanted to answer. One question was about sphingosines and are they made in the body or how do we get them? And they're found in food in the form of sphingolipids, and then they're broken down into smaller molecules that are then absorbed into the bloodstream, and then they are converted to sphingosines in the liver. And then another good question that I was asked, and I've been asked this before, is so we know the carboxylic acid is written COO minus, and why is it not written as CO2? Yes.
Because that's an extra electron, sir?
Yes, it has an extra electron, and it's also a different molecule. So COO, when they're separated like that, that typically means that it's not forming the same type of bond that we see in carbon dioxide. We know that the carboxylic group is typically a part of another molecule, and it's an acid. And CO2 we know is a gas, and of course they have different functions. So that's why we don't see the nomenclature written in the same way, even though they have the same atoms. Does that make sense? All right. So on to nucleic acids. So it's important as physician assistants that you guys understand how nucleic acids work, because they are involved in cancer formation. Our DNA is what's affected either by genetic heredity of a DNA mutation or an environmental exposure to a toxin or a mutagen that's going to affect our DNA, which will then change our proteins and it could potentially cause cancer. Also, understanding nucleic acids is important for understanding shorter term conditions like we know the flu, COVID, all of those are formed by nucleic acid. So understanding how nucleic acid works will be very important for you guys in understanding what your patients are dealing with, and then what tests to request from the clinical lab in order to get the proper results. And then of course you're going to take that information and design a treatment plan for your patient. So we know that nucleic acids, particularly DNA, houses our genetic information. That is what DNA's role is. Nucleic acids, there are two distinct types. We have DNA, which is deoxyribonucleic acid, RNA, ribonucleic acid, and we're going to talk about the different structures of each and the different functions. So hopefully you guys all remember the central dogma theory, and that theory is that DNA, which contains our genetic information, is replicated, and that is replicated to pass that information to new cells. It's also transcribed into messenger RNA. Messenger RNA will then leave the nucleus of a cell and go into the cytosol, where that messenger RNA contains that codon, which is then used to synthesize proteins. And so this is central dogma theory. This is how our genetic information is passed to new cells, and is passed on to give us the function of our proteins that we have based on our genetic makeup. So the components of nucleic acids include the nitrogenous base, which is shown in blue here. This nitrogenous base has two nitrogen atoms that are hydrogen acceptors, which is hence the base, why it's called a base. We also have a 5-carbon sugar and a phosphate. So those are the three components of nucleic acid. So in DNA and RNA, there are two, yes?
And the sugars are always in pentose?
Yes, the sugar is always a pentose. There are two types of bases in DNA and RNA. You have the pyrimidine, which is one ring base. The purine is a two ring base. And as I mentioned, the nitrogens, they each contain two nitrogens that are hydrogen acceptors. So in DNA, the pyrimidine bases include cytosine, shown here in green, and thymine, here this blue color. Those are the pyrimidines, or the single cyclic ring. The double cyclic rings are adenine and guanine, and these bases are found in DNA. Now, in RNA contains cytosine and also contains adenine and guanine, but instead of thymine, which is contained only in DNA, uracil replaces the thymine. So that is one of the differences between RNA and DNA. A little trick to help remember the different components and structural features of the base structures. This little mnemonic cuts the pie. Pyrimidines, pi is coming from pyrimidines, and pyrimidines contain cytosine, uracil, and thymine. Now for the purines, there's two purines, adenine and guanine, and so pure is gold times 2. Pure for the purine, as for the adenine, gold for the guanine, and there's 2 ring structures in purines as shown here. Now both DNA and RNA contain 5-carbon sugar, which is a ribose. The difference is that DNA does not have an oxygen molecule off the 2' carbon as shown here. So hence the name deoxyribose. Now RNA does contain that oxygen group on the 2' carbon. So that's another difference between DNA and RNA. One feature on how the sugar carbons are counted, you notice there's a prime after each number, and the prime is just to signify that you're counting the carbons on a sugar versus the carbons on the base structure. So we've learned up to this point that when we're counting the carbons on other molecules, it's just the numbers. But because we're moved to the pentose sugar of the nucleic acid molecule, we're going to use prime to denote that we're talking about the sugar moiety in nucleic acids. Does that make sense? So I mentioned that a nucleotide contains, or nucleic acid contains, the nitrogenous base, the sugar, and the phosphate group. So when a nucleoside is, the only difference between a nucleotide and a nucleoside is a nucleoside only contains the nitrogenous base and the phosphate, and the sugar. So when the nucleoside interacts with the phosphate group, it forms a phosphoester bond on the 5' carbon of the sugar moiety, and that creates a nucleotide.
Can you say that again?
Yes, so the nucleotide contains the phosphate. The nucleoside does not contain the phosphate. So once you add the phosphate group to the nucleoside, you get the nucleotide. And the phosphate group attaches to the 5' carbon shown here of the sugar component. And this molecule is the nucleotide. Any questions? Here are the different nucleotides found in both DNA and RNA. Remember, RNA contains uracil instead of thymine, and DNA contains only thymine. So if you look at the nomenclature at the bottom, the adenosine monophosphate in black, that represents RNA because there is oxygen found on the 2' carbon shown here, and the deoxyadenosine monophosphate shown in pink represents DNA, and it's showing just a hydrogen atom off of that 2nd carbon, because remember, DNA does not contain an oxygen on the 2nd carbon of the sugar. Any questions? So the primary structure of nucleic acid just consists of nucleotides that are joined together, and they're joined together by phosphodiester bond. And in this bond, we know that this is a nucleotide here where the 5', the phosphate group is attached to the 5' carbon. What this nucleotide does is it also forms a bond with the 3' carbon on the sugar moiety of the nucleotide next to it. And so I've circled the 2 phosphodiester bonds. Here's one of them, and here's the other one. So that's why it's phosphodiester, meaning 2 ester bonds. Any questions about that bond?
So the ester bond, the phosphodiester bond that goes up and down, that one's attached to what carbon? Fifth as well?
The 3rd carbon.
Third carbon, okay, no, that makes sense.
I'm trying to see if there's a better picture. It's attached to this carbon here.
Okay.
And then, of course, the other one is attached to the 5th carbon. And so, phosphodiester bond is similar to an ester bond, except that instead of a carbon atom, it's a phosphate, a phosphorus atom, and there's 2 ester bonds being formed. So hence phosphodiester. So, are there any questions about that? So each nucleic acid has its own sequence of bases, and that is indicative of its primary structure. That's, of course, our genetic information is carried in that sequence. And the sequence is read from the free 5' end to the free 3' end. And it's often written using only the letter of the base involved. So this short sequence of nucleotides is 5', A, C, G, T, 3'. And we're going to go over an example on how to determine the complementary strand of this molecule. So here is a DNA double helix. The purple ribbon backbone represents the phosphodiester linkage between the base molecules, and you see there are hydrogen bonds formed between complementary base pairs. So there are always and only 2 hydrogen bonds formed between adenine and thymine. Those are complementary base pairs. Hydrogen bond shown here. And there are always 3 hydrogen bonds between guanine and cytosine shown here. adenine and thymine are bonded together. Guanine and cytosine are bonded together. So, is someone willing to tell me the complementary base pairs for this sequence of DNA? Yes.
T, C, A, G, G, T, T, A, G.
Correct. Does that make sense to everybody? A always bonds with T, G always bonds with C when we're talking about DNA. Yes.
Does adenine and uracil only have 2 hydrogen bonds as well?
Yes. Yeah. Good question. Good. So DNA is replicated in order to transfer our genetic information to new cells. And we're going to go over how DNA replication occurs. What happens is the DNA molecule will separate, allows for the enzyme responsible for generating the new sequences to come in and synthesize 2 new molecules or 2 new nucleotide strands. So in the new DNA molecule that is synthesized, one of the nucleic acids will be from the parent or original strand, and then one will be newly synthesized. So that is called semi-conservative replication because you have one old, one new. And it's of course going to be complementary base pairs that combine to form the new daughter DNA strands. That's what they're called. So here's just another picture of that. The turquoise ribbons here are showing the daughter strands that are being synthesized. So here are the steps of DNA replication. If you guys go to the next slide, you can see a picture as well, whichever you want to look at to go over the steps. But what happens is the helicase enzyme separates the double helix strands, and then you have the single-stranded binding proteins will bind to each of those strands to prevent the strands from reconnecting. At the site where the DNA strands have separated, that's called the replication fork. This fork is where the DNA polymerase will synthesize the new DNA strand. And so we're talking about the leading strand, which is on the top strand of the picture schematic of the DNA replication. On the leading strand, DNA polymerase produces one continuous chain of a nucleic acid going in the 5', 3' direction. I circled the 3' on the purple strand just to show you that is the end of the original strand in purple. So we know DNA synthesis occurs in the 5', 3' direction. So that's why on the leading strand there's going to be one continuous nucleic acid formed by DNA polymerase. Now the lagging strand is a little bit different. So on the lagging strand you have primase enzyme that adds RNA primers that are complementary base pairs to the lagging strand. That signals to DNA polymerase to add the DNA nucleotides to that region. So basically the DNA polymerase will remove the RNA primers and add DNA nucleotides. Those short DNA nucleotide sequences added by DNA polymerase are called Okazaki fragments. And so then we have DNA ligase that joins the Okazaki fragments together to produce a continuous lagging DNA strand. Any questions about that? Yes.
So the top strand, the polymerase is going
So it's going from left to right only for the primers, but then the ligase is connecting them right to left. So that's why DNA, the direction of DNA synthesis is from 5' to 3' no matter what. That's the overall direction. Any other questions?
I have one, sir.
Yes.
So when you say so 5 to 3, are you talking about the strands 5 to 3? It's like the direction.
Yes, because the strands, remember, they're anti-parallel, so,
Yeah.
so yes.
Okay.
So in this, the top, this is the original or parent strand. So DNA polymerase is gonna go from 5' to 3'. So it's just gonna be one continuous nucleic acid synthesis because it's anti-parallel, so it's 5' to 3'. Now with the lagging strand, it's 5' is the original strand, so the primer or primase is gonna add short nucleotide sequences that are gonna be replaced for DNA by DNA polymerase, and then the ligase is gonna connect them in the 5', 3' direction. Yes.
So the Okazaki or fragments are only on the lagging string?
Yes, the Okazaki fragment is only on the lagging strand. All right, first check on learning. So match the following to their definitions. What is a short segment formed only on the lagging strand? Okazaki fragments. What is the starting point for synthesis of unwound DNA sections? One person, loudly please. Replication fork, thank you, yes. What's the enzyme that unwinds double-stranded DNA? Helicase. And the enzyme that catalyzes the formation of the phosphodiester bonds of complementary base pairs. DNA polymerase? Yep. Any questions? Correct, all correct, yes.
So I just wanted to talk through the direction of replication so I can answer if that's all right with you.
Sure.
Okay, so,
Can you speak up?
so if I wanted to transcribe the written form, not the picture form, right? So it'd start with the helicase. That's what's catalyzing, that's the enzyme that's catalyzing the splicing of double helix. Then 2 would go over single strand binding proteins. And for 3 would be the DNA polymerase, which is coming along behind that, correct? This is traveling from the 5' to the 3' direction. And then, so our lagging strand with the DNA ligase and the Okazaki fragments, is that happening concurrently?
Yeah, they're all happening at the same time, but at different speeds because remember the lagging strand, a lot more is going on than in the leading strand. The leading strand is just replication fork signals, okay, this is where DNA polymerase is gonna start the nucleic acid synthesis, so if there's nothing stopping it from just forming that straight chain, but on the lagging strand, there's a lot more steps to get that nucleic acid synthesized.
Okay. So the lagging strand is half of the strand that is, for lack of better explanations, kind of, it's like a free running end almost.
Is what?
It's like a free running end. Like on a piece of rope, tying it out or something. That's like your lagging strand is like your free running end, while the helicase is working its way down the strand. The DNA polymerase is going on that top one.
Okay.
They're working concurrently. I'm just trying to make sure I.
No, however you understand it, whatever's easy for you to understand this process. So it's just leading strand, continuous formation, lagging strand. It has more steps in order to get the new nucleic acid sequence, and these two will break off to daughter strands. So for every one DNA molecule, there's going to be 2 daughter strands. Any other questions? Alright, so now we're going to talk more about RNA, messenger RNA, and the different types of RNA. Here's an example of ribosomal RNA. Ribosomal RNA combines with proteins and to create ribosomes, and ribosomes are the site for protein synthesis in the cytosol. 3 main types of RNA. You have the ribosomal RNA, as I just mentioned. It's forms of proteins. Then you have transfer RNAs. Transfer RNAs carry the amino acids to the ribosome and transfer that to the growing peptide sequence. And then, of course, our messenger RNA contains our genetic information. So the messenger RNA contains the codon, and the transfer RNA contains the anticodon. So messenger RNA synthesis is transcription, and what happens in transcription, the RNA polymerase uses that DNA template to produce messenger RNA with complementary base pairs to that DNA. Of course, it's going to replace uracil for thymine, because uracil is only found in RNA. And so what happens when that messenger RNA has been processed, it moves from the nucleus into the cytosol to the ribosome for translation, and that's where protein synthesis occurs. And so, as I mentioned, the transfer RNA, it's the molecule that converts the information from the messenger RNA into the amino acid sequence in a process called translation. So when messenger RNA is synthesized, it contains both introns and exons. Exons have our genetic information that needs to be passed on. Introns are kind of junk in the sense. We don't need that information in order to pass the important genetic information for protein synthesis. So what happens is, in the schematic, you see the gene, which is the DNA molecule. It's transcribed into pre-messenger RNA, both of which contain both introns and exons. That pre-RNA is processed, which removes the introns and splices only the exons together. Exons will then leave the nucleus, travel into the cytoplasm to the ribosomes, where they will be translated into the proteins that our body needs based on our genetic makeup. Does that make sense? So our genetic code is contained in triplets, meaning 3 nucleotides will code for 1 protein. We know that there are 20 amino acids, and each of which have multiple codons that code for that specific amino acids. We also have start and stop codons. AUG is the start codon. It also codes for the amino acid methionine. This is the initiation of protein synthesis. Then there are 3 stop codons shown here, and those signal termination of protein synthesis. So if we have this messenger RNA sequence here, remember the codon is in triplet. So CCU codes for proline, AGC codes for serine, GGA codes for glycine, and CUU codes for leucine. So this messenger RNA would produce the following peptide sequence: proline, serine, glycine, and leucine. Any questions about that? Yes.
Are these still matching up? Like the CCU, is that gonna like match up with anything else?
No.
Not
No.
like adenine.
No.
thymine.
No, it's only gonna be, the codons are specific for specific amino acid. Yes.
How much of this genetic code should we know?
Good question.
Thank you.
So you guys have to know this whole table? No, I'm just kidding.
Oh.
Never do that to you guys. So you guys will be given this table, and if there is a question on the exam, similar to what you just saw, then you will use this table to determine what amino acid codes for a particular triplet codon. For example, if we want to determine the codon for valine here, you use the 3 letters. This is the first letter. Here's the second letter. So the second letter contains these 4 letters, and then here's the third letter. So valine would be G, U, U, and you see that valine, there's 4 different codons that code for the amino acid valine. So all of the amino acids have more than one codon that signify that particular amino acid. Does that make sense? Then you have, let's look at a stop codon. So stop codon, one of them would be UAA or UAG. Both of those code for stop codons. Is it clear on how to use this table to determine what amino acids code for a particular triplet sequence?
So wait, so can you, I guess, what would a question look like then on the test? Is that what you're?
The question would look like something like this.
Okay.
So let's see if we can find CCU. So we CCU, and that's proline. Does that make sense? Any other question?
Yeah, I was just gonna ask you to do another example.
Okay.
And that
Was that, do you have it now? You want me to do another one?
that makes it down with this.
Okay. Yes.
But this is only in the context of mRNA, correct? So it's, not to get too into the weeds or ask for like answers ahead of time, but we're not necessarily be expected to go from DNA to mRNA and then.
Heck no. I don't need to know right now. Let's not do that sometimes. You don't need that kind of pain. Any other question? So, can someone tell me the messenger RNA sequence that is produced from the template below? One person loudly, please, from our ears, volunteers. Yes.
Number 2.
Correct. Does that make sense? Remember, messenger RNA, no T's, no thymine, only uracil. Yes.
So the 3 and the 5 in the first, the thing at the top, that's coming from it being from like the 3rd carbon, right? Like this is like all the way from at the beginning of the PowerPoint. That
Yeah, so this is the free 5' end, and this is the free 3' end
okay.
of the
So
DNA molecule. So remember that DNA molecules are anti-parallel, so there's the other strand would be going this direction, 5' to 3'.
because it's going anti, so it's going the opposite direction.
Yes.
Okay.
And of course, it would be the complementary DNA strand. Here is the complementary RNA strand.
So that's from the top one is DNA and the bottom one is RNA.
Yes. Any questions? Does that make sense? Well, as I mentioned, the tRNA is kind of like it helps convert the message from the messenger RNA into the amino acid sequence. So the tRNA contains the anticodon. So what happens is on the tRNA molecule, here's the tRNA molecule. Looks like a clover leaf in 2 dimension, but the 3-dimensional shape is more like an L. In the tRNA molecule, there's the acceptor stem, which is nucleotides ACC. That's where an amino acid binds and is carried to the ribosome to build the protein. The anticodon is here, so the amino acid that binds here codes for whatever anticodon is here, and this anticodon matches up to the codon found on the messenger RNA molecule. Does that make sense? So the messenger RNA contains the codon that is derived from the DNA. The tRNA contains the anticodon, and remember they're complementary base pairs, and so the tRNA, the amino acid attached, will be the amino acid that codes for the anticodon, which of course then will code for the codon to build the protein sequence. Yes?
So are the acceptor stems and the anticodons opposites of each other? I'm misunderstanding.
Say again?
The acceptor stem at the top, is that correlated to be the opposite of the anticodon?
The acceptor stem is just where the amino acid binds based on the sequence here. So the triplet sequence here, if it codes for serine, serine is gonna bind there. Yes?
So with the tRNA translating with the messenger RNA, both of them are RNA, and then it turns out to be DNA afterwards?
No, it's protein. Because the tRNA contains the amino acid, and so it's gonna go to the ribosome where the messenger RNA is. It's gonna interact, find its triplet codon on the messenger RNA, and once it finds its triplet, it's gonna attach the amino acid to whichever the previous amino acid is to build the protein sequence. Does that make sense? No?
Do you have a video?
I don't have video. Videos are so hard to show here because the internet's so bad. But so let me explain what was actually on this side. So this ACC is the acceptor stem. What all that means is that whatever anticodon is here, if these nucleotides code for serine, serine is gonna attach here. That tRNA is going to be transferred to the ribosome, and so if the first codon on the messenger RNA is serine, serine is going to be the first amino acid that starts the protein synthesis, and then the next tRNA will carry another amino acid, and if that is the codon or on the messenger RNA, that amino acid will attach to the first amino acid serine. And then it just builds that process until it reaches a UGA or a stop codon, and that will terminate the amino acid sequence. Yes?
I think one of the questions you might have been just like putting it together, so it's like DNA, mRNA, tRNA, protein. Is that?
Kind of, but tRNA is already built, is already made. So like DNA, RNA, and protein is kind of like one is being made from the other. But the tRNA has already been synthesized and it's just carrying the amino acids to build the protein sequence. Yes?
So is this,
Can you speak up?
sorry, I'm
No,
just
no, it's okay.
So the mRNAs are in the cytoplasm, it's getting ready to process this, the tRNA is coming up and essentially like attaching to the mRNA and actually.
Yes, it interacts with that codon, that triplet codon, and then, so it's gonna be multiple tRNAs coming, and they're gonna, if it doesn't match to the codon, it's gonna be like, oh, and then whatever the one that knows is the correct complementary base pair, or the anticodon on this tRNA right here, whatever one is the appropriate complementary base pair, it's just gonna add. So it's like so much going on at one time to build that protein sequence. Is that Yes.
What's the proportion of tRNA to mRNA? So is there a bunch? Is it like The Bachelor? There's like one mRNA
Yes.
and a bunch of tRNA. I don't want you. I don't want you.
Thank you. I'm gonna use that for next game. Yes, that's a perfect analogy. Yes, it's a lot because you know there's gonna be like it's only one anticodon, and so it's gonna be they're floating around trying to figure out if they can be added on, and if not, they're gonna bounce off, and it allows the actual one that's a complement base pair to add the amino acids. Yes.
Trying to make this make sense, that the tRNA is like a subcontractor that they hire. Finish the bathroom, finish the kitchen,
Yes.
finish electricity, all that stuff.
Yep.
Okay.
Man, y'all, I need to write this stuff down because this will help me next semester. Yes. Hopefully that's starting to make a little sense. Yes.
There's a unique tRNA like structure for every anticodon combination.
It's gonna be, this is gonna be the unique part, the anticodon loop. So that anticodon loop is gonna be the signal for the amino acid to attach to the acceptor stem. Does that make sense? Hopefully a little bit better. Yes. Did you have a question?
Yeah, so the anticodon is pretty much dictates what gets attached to the acceptor stem, and it's not always going to be ACC, it can be
The acceptor stem is always gonna be ACC. That's just the sequence of that region. But you're right, the anticodon determines what amino acid attaches to this tRNA. Any other questions? Yes. Can you speak up? So this is just in the cytosol. The amino acid will attach to the acceptor stem based on the anticodon loop, and then it will travel to the ribosome. And where the messenger RNA is, because the ribosome is like kind of helping all this, the attachment come together of the amino acid sequence or the protein sequence. Does that make sense? Any other questions about this? Yes.
Can you think of like the ACC is just like if there was like people are forming a line and there's like a flag at the end of the line, like where it's gonna be going next, that's just like flagging it for the codon to be added, right?
That's for the amino acid to be added. Yeah, that's another good analogy. It's just a site that says add here for the amino acid, but it's gonna be added based on that anticodon section. Any other questions? Yes.
So once the translation complete, does the amino acid they just like detach, or they still bond together?
The amino acids are being attached to each other by another enzyme called amino acyltransferase. That's forming and you don't need to know that, but that's forming the amino acid chain. And so once the stop codon occurs on the messenger RNA, that's going to stop the protein synthesis, and then the polypeptide is going to detach from the ribosome and then start doing its folding or whatever it does to produce its function. Any other questions? You guys take a 10-minute break.
Just take
It's like a representation of why like how fast freaking nucleotides are getting pulled in to build it. And you're like, oh, okay, so this is not like a so one's going this way and the other one's going this way. Chinese So like you have your DNA, right? Beautiful DNA, whatever unwraps, you have three, and nothing else comes out. Right? This side, we'll call it a side. This side is okay, so that's the 5' that one or whatever, right? So when this thing is like getting this side of this everything's making is taking this code thing behind it and it's working on it's doing it right? So this guy can only do it by 3. And for whatever reason, the extra little starts. He has a bunch of those starting transfer and hopping down the line. All it's going to do as it A, B, and A are going to find the starting points and then just complete it. Right here's your 5 side. This side is just going to be this way. And it's just going to take this one down next to while it's doing that. So this goes up here. I don't think only move in one direction, whatever it's right? So that primate is going to so yeah, you can think right now. Separates that first so the red part, that first replication is going to look like that, right? You're going to have 2 pairs of strands, 2 straight, right? But then when that goes down to the next level, you're going to look like this, and then it's going to look like, I guess it goes to and also have my you know what I mean? Over here, that will look the same. So you start losing that original top for that line, really that deep bit as you're changing, right? And that will look the same. So you'll have like like by the next layer of reputation, you're really gonna have 2 years. So all this stuff is just a bunch of. Yeah, and that's the inherent change.
I feel bad when I'm
It is
just always get it the first time.
So think of it like right, so I'm gonna write it. ACC, right? That's what it looks like, right? 5 times 3 times. No, the strand, you gotta take the sin of the model. You have this like 3 or 5 primes. Right? Go back to like your maybe if you have you have the 5 primes, right? You have that like 3 thin particles that's just kind of floating out. It's like a very thin, right? The bottom side of that 5 prime, you have the 3 prime part. Just the third part of it, that is the opposite. Right? That's the 5 prime part you're talking about. That's
Only the difference between
so if you think about it, and you start connecting down the street like this. These are gonna bond like this. Right?
He knows how to do it.
Well, yeah, technically they're but in the general scheme of view, this 5 is top. This is going to be the bottom, right? So we're seeing
She loves the G. She loves it. She's like, yeah.
and 5 prime down here, so that they can come in the middle of very
Yeah, viruses are fun.
5 on this side. Imagine there's like 1,000 of these all connected. And that finally is just ending one. Start with 5 prime on the side, ending one is going to end with 3 prime. And then this side, yeah, it's start with 3 prime. And as you connect thousands and thousands of these together, more like beads, that's 5 prime. Oh wait, we'll figure it out. Oh yeah. That was the part because they all had
I don't know if it's on the
Alright guys, let's get started back. Are there any burning questions about translation? Any other questions? If you think of any, please ask me next class tomorrow or Canvas any questions you have about this. And so this picture here is just showing how an amino acid attaches to a tRNA at the acceptor stem, and it does that by an ester. That's the point. And of course, as I mentioned, the amino acid that binds to a tRNA will be based on this anticodon loop. anticodon loop interacts with the codon, which is found on the messenger RNA. Okay. So, if there is a genetic mutation in our DNA, it's going to cause a number of different things with varying severities. Here we see an example of peacock that has albinism. You know, peacocks are normally very colorful. They're very mean animals, but they're very colorful and beautiful. But and when there's a genetic mutation in their DNA, they can have albinism, and we're going to talk about how that occurs. And so a mutation is a change in a DNA nucleotide sequence that may or may not result in a change in the protein that is made during the translation process. And so these mutations can be passed on from in our heredity, or also if we have some environmental exposure to a toxic agent, we can also get a genetic mutation in our DNA. And typically that is when we see cancers forming. And so you can have a range of severity of conditions, either a disorder or disease, or and sometimes the mutation is lethal, meaning that typically causes a premature stop of the protein synthesis. And so that means that typically when that happens, if it happens in humans, the human is not going to survive for long periods of time. And so here is a normal DNA sequence. You see the DNA template strand transcribed into the messenger RNA, which is translated into the amino acid sequence with the stop codon. So there are 3 different types of mutations that we're going to talk about. We're going to talk about the point mutation. Point mutation, there's 3 types of point mutations, excuse me. A silent mutation is when there's a change in a nucleotide. However, that change codes for the same protein or the same amino acid. So if we take this example, CAA is the codon sequence in the is the nucleotide sequence that codes for glutamine, and CAG, that last A is changed to a G, that also codes for glutamine. So in this case, that's not going to change the amino acid or change the protein in that sequence. So this is called a silent mutation. A missense mutation is shown when one amino acid is changed and it codes for another amino acid. That same, looking at that same glutamine example, CAA, if the nucleotide is changed from A to C, that middle nucleotide, glutamine will then be code for proline. So, can somebody tell me what type of amino acid glutamine is based on the functional group? Polar neutral? All right. What about polar? Say again.
Non-polar.
Non-polar. So, great, you guys are correct. So, these 2 different amino acids will affect the type of tertiary bonds that we talked about. Remember the 5 different bond types: hydrophobic, hydrophilic. They are very different. The R group is very different in function, so that's going to affect the folding of the protein structure. And so therefore, in a case like this, of course it would depend on the particular protein, but that can make the protein less active or even inactivate that protein, whatever the protein is being synthesized. Yes? Say it again.
Where would the R group be at that example?
This right here, these are the R groups. So if we have glutamine, it's supposed to be the amino acid, and it's a mutation causes it to be changed to proline. This is a missense mutation. Then you go from this R group to this R group. This R group is non-polar. This R group is polar. This R group is non-polar. So that's going to affect the type of interactions that we saw when we looked at the tertiary structure interactions. So that's going to affect the protein folding and things like that, and the 3D structure. Then you have the nonsense mutated codon, and that occurs when the change in the amino acid, the change in the nucleotide sequence codes for a stop codon. So that is typically going to be more serious because the protein amino acid sequence is going to stop prematurely. You also have frame shift mutations. Frame shift mutations either adds or deletes nucleotides, and in that sequence, and remember, nucleotide sequences, it's a triplet codon. We read them in frames of 3. So that's going to affect what amino acid is synthesized based on if there's nucleotide added or removed. And here's an example. So if we remove a nucleotide, it's going to shift the reading frame, and it's going to change the subsequent amino acids that build the protein during translation. Any questions? So there are a number of diseases that are due to frame shift mutations, and they include cystic fibrosis, Crohn's disease, and familial hypercholesterolemia. So here's an example, well, actually 2 different examples of genetic diseases that occur from proteins that whose functions have been reduced, and this is one pathway. And so remember I talked about how phenylketonuria is commonly talked about in biochemistry. So what happens in phenylketonuria, this is phenylalanine, the enzyme. It's converted to tyrosine by phenylalanine hydroxylase. In phenylketonuria, this phenylalanine hydroxylase enzyme is not functional, so that's going to cause phenylalanine to be converted to phenylpyruvate and phenylacetate. Phenylpyruvate and phenylacetate are toxic to the central nervous system, so that will cause mental delays and mental disabilities in children that have phenylketonuria. So there are some treatments that you would give to those children, and we're going to talk about that in future blocks. But also, in the same pathway, as I mentioned, phenylalanine is converted to tyrosine by phenylalanine hydroxylase. Tyrosine is then converted to melanin. The enzyme that does that is tyrosinase. If tyrosinase is non-functional, then tyrosine will not be converted to melanin, and that will cause albinism. Are there any questions? So here are several genetic diseases caused by mutations. If I were you guys, I might want to know these diseases and what causes the genetic mutation. You don't have to know the incidence numbers, you don't have to know every detail, just know the specifics. For example, when we look at Down syndrome is the result of the formation of 3 chromosomes, trisomy, usually number 21, and we know that causes mental retardation, right? Also, Tay-Sachs disease, another example. There's a defective enzyme, hexoaminidase, which causes accumulation of gangliosides. Gangliosides are glycolipids. Once the gangliosides are broken down by this enzyme, but if this enzyme is defective, our brain will have a buildup of gangliosides, and that is kind of toxic to the brain, and that will cause early death in patients that have this accident. Are there any questions about this lecture?